Yeah, so the next thing that we're going to be talking about today is the FoldTree. It's a really important data structure in Rosetta. It's pretty powerful. And you'll get to work with the FoldTree and get to sort of experience firsthand what types of effect it has in different flavors of modeling or different tasks later on when you're doing the lab today. So just a quick overview of what we're going to go through. First, we're going to talk about some of the motivation for the FoldTree. There are lots of different motivations that you can talk about. I'm probably going to just talk about one of them. About a little bit about the data structures, the FoldTree, the atom tree in depth, how you can use the FoldTree and interact with it to get different types of modeling in your protocols and different benefits. And then finally, as I said first, we'll do a lab where you'll get to actually chop a protein up into secondary structure elements, model this in the FoldTree, and get to sort of play with what type of effect it has. If at any point in this lecture you get confused or stop me, just feel free to ask questions and interrupt. This is a cool, fun lecture and want it to be interactive. So one of the motivations that I learned about the FoldTree with is actually in the ab initio folding application. If you're not familiar, the way, or at least the general way that we do folding in Rosetta is by walking along an unfolded structure and inserting 9 and 3 residue fragments, which have a set of backbone torsions to them. And then what you'll do is you'll update the torsions using those fragments and then evaluate the energy of that particular fragment move. So did inserting that fragment make the structure beneficial or not? But what's a really interesting effect of this fragment insertion process is the effect that it might have when you already know the secondary structure, which I believe is really important for ab initio. So let's say I were to have just a simple structure and there are 3, it's like 9, let's say you're inserting like 3 residue fragments, so it's 9 residues long. And fragment 1 and fragment 3 represent different beta strands. If I were to walk along and change fragment 2, and I were to insert it and update the coordinates of fragment 2, because we work in internal coordinates, we can figure out where fragment 3 is going to be. So this is if you're sort of walking along the structure and updating it from N terminus to C terminus. But an interesting sort of crazy effect that you have is that if you update the structure in this way, then you'll break the long-range interaction that might have been necessary between fragments 1 and 3 to form the beta sheet. So this is sort of an interesting problem that I believe was sort of addressed in Rosetta by the FoldTree. Because you commented this question, how do I update the coordinates? Yeah?
Just to make sure what I you know that the data structure of 1 and 3.
Right. So the question was, do you know the secondary structure? And the answer is yes. When you run ab initio, I believe you provide secondary structure prediction.
So probably this fragment insertion is based on sequence rather than knowing a couple of secondary structures.
Right, so I think regardless if you're inserting fragments and you're trying to determine the secondary structure from that, you'd still have the same problem, whether you provided secondary structure when you're generating fragment libraries or not. So I think the same, whether you provide it or not, I think you still have the same issue, if I'm correct. Any of the instructors, feel free to correct me on that issue.
So usually you use the full sequence to predict the secondary structure.
Right.
And then and that's only so accurate. So then you measure best to say maybe there's an 86% chance that this will be helical, and the other 15% is going to be beta sheet. And so your fragment set will, for a particular structure, have both helical fragments and one beta sheet fragments. So that would be one of the inputs that we go into ab initio.
Right, right.
We're going to go into the fragment picking before ab initio.
Right, so, but the issue that you're sort of trying to address here is that as you insert those fragments, so I'll talk about this in a couple of seconds, but the challenge in saying that there's some sort of long-range interaction that forms a sheet that's not as easy to predict as, say, in like a helical secondary structure. So yeah, this is an important question. How do the coordinates get updated, and how can you walk through the structure to figure out how conformational change should propagate when you make a move in the structure or you change something about the structure. So the first thing that's really important to know in this talk is Rosetta uses internal coordinates. This means that we're representing, or at least we're working with the structure in a series of dihedral angles. The first atom in the system always need to be represented in Cartesian coordinates. But what's good about internal coordinates is that you can update the coordinates of the next residue based on the coordinates of the previous. So you can sort of walk along the backbone and update your dihedral angles when you make a change. And this has some important effects. This means that if you, when you make a change in one residue, they propagate downstream. And then second, if you make small changes in internal coordinates, you'll result in larger changes in Cartesian coordinates downstream. So the question is, how does this, oh, Andrew, I guess the slide change didn't get updated. It's okay. So the thing that you want to start to think about is when I make a change in the coordinate in Rosetta, so like in the lab last night, when you say set by, what happens? So Rosetta actually uses this really cool algorithm that does sort of recursive refolding. Recursion should sound really familiar, like in the homework assignment that you did by computing the recursive Fibonacci sequence. And what you can actually do is if you were to represent, start thinking about if you were to represent the all-atom version of your structure as a tree, just kind of like the graph we talked about this morning, you could actually recursively walk the tree and start to update coordinates. So let's say here that the red residue represents the N-terminus and the blue represents the C-terminus atom. And we'll talk about what these arrows mean in a couple of minutes. What you can do is you can start at that, calling it the root. And let's say you were to move the N-terminus. If you were to call, you could then call update XYZ coordinates, which would then update the children of that particular root atom. So every atom that red vertex is pointing to. And what you could do is then recursively walk the tree, continually traversing and updating the coordinates of your children. And as you walk along the structure, what you've then accomplished is that because you're working in internal coordinates, you've used the change in that first position to update the internal coordinates of the rest of the residues. And this is sort of how when you make a change in Rosetta, you can start having that change walk along the tree. And this is just some pseudocode of what the recursive version you would look like. You would call update XYZ coordinates. Mary? Yeah?
Does that always propagate downstream? Like, do you always start at the root, and even if your change is like at the tail end, it just starts at always?
So the question is, does the change always propagate downstream based on where the root is? So this is something that we're going to talk about in a couple minutes, how you might want to change that and benefits that you can get. You can change the direction of some of those arrows. The important thing to know is that wherever you start, where you go next is going to depend on the direction of the arrow. So let's say in the red vertex, if one of those arrows were flipped up, you wouldn't change the one above it. You would only work downstream from it.
So I think maybe Steven's question was if your change is pretty close to the C-terminus,
Right.
you really walk all the way from the N-terminus down to the C-terminus. Like, that is, if it's really far away from
Right.
the root, is
Okay.
there a faster way to go?
Yeah.
Sorry.
So in this case, I just sort of decided that the place that I was going to make my change was at the root. But I believe Stephen is correct, where if you were to make your change closer to the C-terminus, it would propagate from the point that you changed. I don't think you would have to update all the coordinates upstream. So yeah, does that answer your question?
Yeah.
Okay. No, you should propagate downstream. So downstream from the one you changed. So if you make the change, I guess it's difficult without a pointer. If you make the change, say at that blue point, that's pointing to 2, if you were to make it at the C term, I guess not literally the C term, but if you were to make it at that blue vertex, it would update the ones below it, but not anything up above it. So only the one that's pointing down towards. So that's kind of part of the motivation of the FoldTree that, like, we'll talk about in a second, where you can sort of start flipping some of these errors and adding them. Because yeah, that's the issue. Imagine that, like, that's the end of a beta sheet and you update that and it pushes that way. You've broken your long range interaction. So yeah. Are there any more questions on this before I move on?
So if you only propagate one way, but you flip that and you introduce a class of that's just going to change. That doesn't matter. Right?
Can you repeat your question? Sorry.
So like if I randomly perturb the end of the tail and
Right.
I end up reflecting the first part of the tail, that doesn't matter in terms of the whole thing.
If you so the question is
So
if,
like say I pick the bottom half of the music and fold it in half and then touch the class at the top. It doesn't matter because it's just folded right now.
I don't believe so. Yeah, the question was if you perturb the bottom of the structure and that results in a clash in the top, it won't change the top after it's updated the coordinates, even though there's a clash. I think that would get resolved in the next iteration of your Monte Carlo where it would score it and realize that there was a clash. You're just sort of in the point where you've just updated the position. It hasn't been scored yet. So yeah, sorry.
So would it be accurate to say that you're using everything upstream in the reference frame and then moving downstream?
The question was, is it accurate to say everything upstream is the reference frame? I'd like to say yes, but I'm not sure. Andrew?
Yeah, I mean, if I understand correctly, so you're saying if you're trying to pick up the coordinate of an atom, you use a frame from its ancestor to help figure out where it should go. And that's true, yeah.
Okay, great. So I'm gonna move on. Cool. Yeah. So this is kind of what's happening at the very low level, but keep this sort of idea in mind as we walk through and talk about some of the changes that might happen to this guy. So yeah. So this is a problem. You could sort of see in that example. You guys touched on it. You know, what if you change the bottom and that resulted in a clash, or you broke an interaction that might have been important to the structure. This is really difficult to predict. If we're looking at helical structures, it's much easier to figure out, say, where a helix might curve, because we know that like every 4 residues, you have a hydrogen bond that will form in sort of that backbone of an alpha helix. But if you have a beta sheet and you're at a particular position, predicting where that hydrogen bonding interaction might form might be a little bit more difficult because you don't have the capability to know that when you're updating your XYZ coordinates if you're just moving in that way, kind of downstream along the entire structure. So at one point we said we need a way to solve this problem. We need a way to predict some of these long interactions, correct for some of these different changes. And this is kind of one of the really cool motivations for the FoldTree and for the atom tree. So the FoldTree and the atom tree are 2 data structures in Rosetta that are really important for describing how to propagate change through a molecular system. So maybe instead of moving downstream, you'll make jumps, which we'll talk about, or changes so that you can update confirmation of a beta sheet. All of this code that I'll describe in this particular lecture lives in core kinematics. This is sort of the internal coordinate library of Rosetta. And what's really important to know about both the FoldTree and the atom tree is that they're describing the geometric connectivity of a particular molecular system. This has some consequences because the FoldTree is a directed acyclic graph. That means there's no cycles. So if you're thinking of on the atom level, if you have a residue with a ring system, so like a phenylalanine, that ring is not, the closure of that ring is not necessarily represented in the FoldTree or in the atom tree. Or in the FoldTree level, if you have cyclic peptides, like you can't necessarily close that. Yeah.
Can you use like a virtual atom to make something cyclic in the FoldTree? Is that
So I don't think you could use, because a virtual atom is still added as a vertex to the fold tree, so I think you would still have a cycle. But we do other things with virtual atoms that we'll talk about later in this lecture that help you to resolve this issue of like, how do you close cycles that are actually there? Oh yeah, the question was, can you use virtual atoms to resolve the cycles? And the answer is, I guess it depends. So yeah, so this is a consequence to think about. These are definitely issues, and it's important to know about them when you're constructing fold trees. So what are the components of a fold tree? So a fold tree is the residue level of this sort of graph that's describing the geometric connectivity of a pose. And the vertices are naming residue positions. So let's say I have one long chain that spans from residue 1 to residue 100, and I just want to have a default fold, a regular fold tree from N to C. My first vertex would be called 1, and my last vertex, the downstream vertex, would be called 100. These aren't like it's not referring to the actual indices. We're just naming the vertices by the indices in the pose. And then there are two types of edges that you might work with in the fold tree. The first is a peptide edge. Peptide edges work for all types of polymers that could be in the pose, so DNA, RNA, protein. And the polymer edges, or the peptide edges, represent a contiguous span of residues. This means they should be consecutive as you sort of walk along the chain. These edges have a special name. You can either, when you construct an edge, you can refer to them by -1, or you can call them core kinematics edge peptide. Is sort of the enumeration or name that you can use to construct a peptide edge. One second. And what's important to know about peptide edges is that residues in the intermediate must be polymer residues. And the higher numbered vertex of a peptide edge is a cut point, which I'll talk about in a second. You had a question?
Yeah, so if you have like multiple chains in one pose, does that mean you have multiple fold trees, or is that always in one fold tree?
So the question was, if you have multiple chains in one pose, do you have multiple fold trees? There's always one fold tree for a pose. It's actually a rule of the fold tree that it must describe the entire pose, so none of the residues can be left out. So the way you do that is actually through the type of edge that I'm going to talk about next, so hold on one second. But yeah, just going back to finish up peptide edges. So peptide edges are sort of this, they're describing just a continuous span of residues, and then you have this thing called a cut point, which is always the higher numbered end of a particular vertex. So if you have any, just for example here, if you have like one beta, let's say your entire structure is like one beta strand, your peptide edge would be from the beginning to the end. So the next type of edge that's used in a fold tree is called a jump edge. A jump edge is a special type of edge because you can relate two local coordinate frames. So examples of this would be if you had two chains that weren't necessarily continuous, or if you had a ligand in your system that you want to relate to the rest of the structure. So this will connect parts of the molecular system that aren't necessarily chemically connected by a polymer bond, but you would want to relate to one another in modeling. My favorite example of this is if you're doing protein-protein docking, and you have two partners, those two partners will get related by a jump edge in the fold tree. So we name the jump edges using integers, so consecutive integers, so just 1, 2, 3, and the number of jumps you have in your fold tree is always equal to the highest numbered jump edge. And then when you add your jump, you'll specify a cut point. So an example of like a particular jump is between the two beta strands to represent that, like the geometric connection. The big idea that we're sort of talking about here is, so how did the fact that now, instead of just walking along the fold tree from N to C, how did I change the way that I'm folding my structure? How did I change the way that I'm representing coordinates? So this is sort of a fun way that we're representing the full tree here. So A is the root. That's kind of that top point where you would start. The arrows are representing beta strands. From the root to the cut point is one peptide edge. And then from the cut point to the end is another. And then that red arrow is representing the jump between the two beta strands. So if you can sort of remember that picture of the atom tree, the picture of the graph that we walked along before, and how you just walked linearly, think about how that would change if you were to update the coordinates using this. Where let's say if you were to make a change at B, you would update the second sheet as well as things downstream of this point. So the question was, do you have to manually reset the cut points? So the cut points are things that when you construct the full tree, you either decide on where they are going to be, or for like a peptide edge, it's kind of like a default definition. I think updating the full tree is I think updating the full tree while you're modeling is not something that's necessary. I don't think you change the full tree topology while you're modeling. I think it's something that you create at the beginning. So I'm not sure. Andrew?
Sometimes people will take
Okay. Okay.
If you have a full tree and you want to update it, let's say you start with an edge from A to E, and say you want to add a jump edge to this full tree. Then you can call a function of the full tree, or maybe it's a utility function to add an edge between E and D, add a jump between E and D. And then when you do that, then you have to specify the cut point. Does that answer your question? Yeah.
Okay, cool. Yeah. So, and this is, so all of the code for the full tree lives in the full tree class in core kinematics. And this is just a quick overview of the interface, or like the most common methods that you might call from the full tree, but I'll go through these individually in a minute. Two main ones that you'll be adding later today is add edge, where you can add an edge to the full tree, or new jump, where you can add a new jump to the full tree. The reorder method is useful if you want to update the directions of your edges all at once. Check full tree is how we know all of the rules of the full tree are kind of followed when you make a new one. And then simple tree will just create a simple full tree from residue 1 to the total residue. So just going through these methods. So the simple tree method, as I just said, this will just create a simple full tree going from the first residue to the last residue in your pose. So this is just sort of an example of like a simple full tree topology from N to C. The next interface you'll find is particularly useful in the lab later today is adding an edge. When you add an edge, you specify the start residue, the end residue, and then the type of edge that you want to add. So if it's a peptide edge, you can add core kinematics peptide. If it's a jump edge, you'd specify the number of jumps. So a lot of times using a jump counter is useful in this context. Another way to add a full tree edge is by specifying new jump. New jump is you can specify the first jump position in your system, the second residue position, and then the position that you would want your cut point to be located at. Next method is reordering. So something really important to pay attention to in the full tree is that the order of the edges matter. As you saw when we sort of talked about propagating change at the beginning, if the arrows are pointing in the different direction, change might not have propagated in that direction depending on where you were. So reordering is something you want to do if you say you want to change the root of your full tree and then flip all of the edges pointing downstream away from that edge. So this is just an example of what would happen if you were to do a reorder on these. If you had your root at the red position over there, and then you were to change it here, the direction of the edges would flip so that everything's pointing away or downstream from that particular position. Yeah?
So the root that you give it in the parentheses, you're defining the residue.
Yeah, so the root is the residue number that you want the root to become. when you would call that method. Yeah. And then, so the FoldTree can be a little bit finicky. So it's really important to know about some of the rules that govern why you might want to add or change an edge. The FoldTree must be a fully connected graph, and every residue must be included. So that means that, as I mentioned earlier, you can't just leave out residues in the pose. You have to include everything in your FoldTree. No cycles. It's a DAG. Cycles would the FoldTree would break. The order matters. Folding or updating happens along the edges, as you got to see earlier. And then finally, a really important method to check for these rules is check FoldTree. This will return false if anything is sort of going amiss in your FoldTree. If you've broken any of these rules, or sometimes there are little things about the order that you add your edges. So it's a really important method before you do anything to make sure that you're doing things right. And so this is an example FoldTree that we just kind of talked about earlier. So I guess we can sort of go through it. So first, like, where would I add my peptide edges here? Okay. Yeah, so I do have it labeled. That is right. But what positions would it start and end at?
I think you would want it to start at 1 and end at 30, and then maybe start at 30.
Right. Yeah. So yeah, that's not quite right.
I guess I'm having trouble with when the position from 30 to 40 would be updated.
Right. So you're definitely going in the right. So the thing was having difficulty with like what happens from 30 to 40. Yeah, that's going in the right direction. Think about like what different peptide edges you might add to fix that problem. Anyone else want to? I'm sorry, can you repeat that? The question was for every different secondary structure, do you have different kinds of peptide edges? You don't have a different type, like a peptide edge is a peptide edge. But yeah, you might want to define different peptide edges to define your secondary structure, your FoldTree. This is also something you'll be doing later, so yeah. Right. Yeah, that's the right idea. Oh, yeah?
So would you want to set the root there at 30? So, because otherwise it would, or would you, do you have to have the root earlier? Or is it just a bad idea to have a cut point at the root in general?
So, no, I don't think you would want to have the root at the cut point. The root is sort of like where you're starting your folding. So like if you were to fold from N to C, the cut point is discontinuity. Also keep in mind that like between those two beta strands, there's probably a loop.
I guess I'm not sure exactly what the cut point is.
Yeah, so the point of the cut point is to say that like, because if you add this jump edge, think about, actually, yeah, I can draw this.
Are you saying that this, like,
Yeah, so,
be at different places?
no, so like, think, yes?
You'll have a gap.
Right. Right, because think about if you have,
You can do something.
Right, and then the jump is actually a torsional connection.
right. Right, so that's correct. So the jump is relating the two edges together. So it's not like you have a hole in your structure. It's just you're geometrically relating it at a different point. So if you didn't have the cut point, you'd have a cycle in the graph. So that's, yeah. So that's where you'd add the peptide edges would be from 1 to 25, 26 to 30, 30 to 40, and then 41 to 50. And then I guess this might be like, yeah. Oh, sorry, yeah.
So would the strand 30 to 40 update in reverse direction starting from 40?
Yes. Yes.
And
Yeah. And I can do. Would it drawing it help? Okay. Cool. Yeah. So the question is, so it's good to think about the directions that these edges might be pointing in, which this diagram doesn't necessarily reflect. What she said is, do you need to flip 30 to 40? Like, is it pointing that way? Yeah, you need to flip it to fold it along correctly. Andrew?
So the peptide would be from 1 to 25, and then from 25 to 30.
Okay.
Not 26 to 30. And then similarly, it would be, say, 40 to 30, and then 40 to 50.
Right. Okay. Yeah. So those should be flipped.
I misspoke again. 40 to 31. So that there's a gap now between residues 30 and 31.
Right. So yeah, the edges that you would want are 1 to 25, 25 to 30. I think it's 40 to 30, right?
40 to 31.
And then 41 to 50.
40.
40 to 50. Okay. Sorry. Yeah, because your cut point is at 30. So. Yeah, sorry about that. Yeah. So are there any questions, or you want to keep going? Okay. Yeah, and then where would you add the jump edge here? Anyone? I'm sorry, what? Speak. Yeah, from 25 to 40. Cool. And so this is how you use a FoldTree with the pose. To get the FoldTree from the pose, you can just call pose.FoldTree. This will give you a const reference to a FoldTree. To set up a new FoldTree in the pose, you can just you can call pose.FoldTree and then set FoldTree in. To show the FoldTree, this will print out the list of edges that describe your FoldTree, and then whether they're peptide or jump edges. You can just do FoldTree.show. And then an alternate way to do this is you can actually just print the FoldTree to standard out, and this will also print the list of edges. And then just sort of one of the last things I'm going to talk about that I want to talk about at the beginning is the atom tree. So the FoldTree is the residue level description of the kinematics in the system, but the atom tree is describing it atom by atom. This is also a directed acyclic graph. It's the same idea. You have one vertex per atom. Bonded atoms represent chemically connected components in the atom tree. And then jump atoms represent, just like jump edges in the FoldTree would represent geometrically related components of the atom tree. And yeah, just to kind of put it all together, all of these components, like what the peptide would actually look like, the FoldTree and the atom tree, they're all sort of giving Rosetta a big picture of how to describe the kinematics in the system as you want to update the coordinates in your refolding. Yeah.
So at the chain break in the last example that you showed, in
Yeah.
the atom tree, that connection will remain between residues 30 and 31?
Can you repeat your question on this step?
So when you have that cut point in
Yeah.
the atom tree, would that connection remain between residues 30 and 31?
No. So the atom tree is also a geometric description, not a chemical one. So it would mirror what the FoldTree would do. Yeah.
so the cut point is necessary
Otherwise, you'd have a cycle in your fold tree.
So at the end of your protocol, does the cut point get reconnected once the close is updated?
So,
Or does it work, or is it like not necessary?
I guess that I mean, I guess like you can update the fold tree and change it, but I think there's other ways to like close cycles, which I'll talk about in a second. Yeah, is that okay for now? Okay, cool. Yeah, so this is just sort of one of the additional steps. One of these unresolved issues that we haven't really talked about yet is how do these cycles get resolved in if you're not describing them in the fold tree or the atom tree. One way to do this is you can add chain break variants. So you add virtual atoms at sort of the discontinuity in your graph. And then if you can turn on chain break energy, this will force the virtual atoms to overlay, which will sort of implicitly try to close the cycle. And then the other thing to know about is we talked about the move map yesterday a little bit. That's what tells the minimizer what parts of the structure can change. So when you're working with the full tree, you want to set jump to true, so you can also minimize over the jumps when you're working with the minimizer. And yeah, so earlier I talked about like ab initio and how some of the basic refolding applications, but the full tree is really powerful, and at least I've noticed it's becoming more and more powerful in trying to fold different types of structures in Rosetta. This is sort of an adapted figure from Phil Bradley's paper, which describes how different fold tree topologies can help you do different types of folding. So my favorite example is in docking. If you want to relate two separate structures or two chains and you want to dock them together, you can do this by adding a jump from the center of mass of one structure to the center of mass of the other to relate the two. And that's just sort of one example. There are lots of others, and you also get to play around with it in the lab a little bit later. So yeah, so now you guys are going to get to do a lab. You're going to take the code that you used yesterday and build upon it. You'll chop the ubiquitin into secondary structure elements and try to model this in the full tree and see how this will have different effects when you model the structure. And does anyone have any more questions? No? Okay.